Alvin Gurulia to explain the research presentation guidelines and mechanics, as well as the introduction of our panel of evaluators. Sir Alvin, you're still muted, sir. Okay, please let us allow Sir Alvin to use the microphone.
Ay na po,
I
sir.
think, okay, yeah, that's it. Not yet pa daw po, Ma'am
Hello.
Zell.
Okay na po.
Loud and clear, sir. Please go ahead. Thank you.
Okay, wait lang po. Okay, so good morning po sa lahat and welcome po sa ating research presentation for the second day of our convention. And I am Mr. Alvin Lulia and I will be the one to present you the mechanics and criteria for judging. As next slide, please. Okay, so for the best research paper, this will be the mechanics and criteria for judging. So papers will be judged by at least 3 experts. The submitted poll papers were sent to the judges before the convention, thus the papers are prejudged. So the best research paper award will be given only to one group, and the winners will receive a certificate of recognition. And the decision of the judges will be final. So for the criteria of judging, next slide please. So this will be the criteria for judging for the best research paper. So we have content and organization, we have 35%. Impact and significance to the community, we have 30%. Originality and technical nobility, we have 20%. And manuscript and language use is 15% for a total of 100%. And for the next one, let me read to you the mechanics and criteria for judging for best oral presenter. So the judging for best oral presenter will be during the convention, and at least 3 experts will judge the research presenter. So the same set of a panel of judges will be judging for the best oral presenter. So it is recommended that the presenters wear either a business attire or school uniform, and the presenters are given 10 minutes of pre-recorded video presentation and 5 minutes to answer the questions from the judges. So every after the presentation of the participants, they will be given 5 minutes for Q&A portion. And the Best Oral Presenter Award will be given to only one research presenter. And the winner will be receiving a certificate of recognition. And of course, the decision of the judges will be final. And for the criteria for judging, we have 40% for video presentation. This will be comprised of the following: clarity of voice and visual, 10%. Organization of presentation, we have 10%. Compliance to time allocation, we have 5%. And visual impact of the video presentation is 15%. For oral defense, it's 60% of the total. It's comprised of voice, confidence, and style, which is 15%. Clarity and consistency of ideas, 10%. Ability to respond to questions, 20%. And ability to convey research findings, 15%, for a total of 100%. So now let me present to you the panel of judges for this year's research presentation. So our first judge is the PSM National Division Representative for Industrial Microbiology. She is also the plant hygienist and quality assurance compliance manager of Interfield Laboratories. And as far as I know, she is also a alumni or alumnus of the Partido State University. So our first judge is Ma'am Kathleen B. Pahoyo. Good morning po, ma'am.
Good
Our,
morning.
good morning, ma'am. Our second judge is the PSM National Corresponding Secretary. He is an assistant professor, a member of Microbiology Unit, Department of Biology, College of Arts and Science of the University of Philippines, Manila. Our judge number 2 is Mr. Edison J. A. Pagoso. Good morning, sir.
Morning.
And our third but not the least judge is the PSM National Liaison Officer. She is also a university researcher and the director of UP Los Baños Museum of Natural History. We have none other than the beautiful Ma'am Marian P. De Leon. Good morning, Ma'am Marian.
Good morning, sir. Good morning to everyone.
Good morning po. And now let me present to you the order of presentation so that we will be guided kung ano po yung sunod-sunod na ating maririnig na presentation. So this will be our order. So the first one to present will be the group of Mr. John Patrick F. Pasibe of Partido State University. Second will be the group of Ms. Honey Joy M. Macalia of Bicol University Pulangi Campus, followed by the group of Mr. Michael M. Calendacion of Partido State University. Next will be the group of Maria Teresa Villaviña of Bicol University Pulangi Campus. The fifth to present will be the group of Mr. Dexter Y. Borilla of Bicol University Pulangi Campus. And the last will be the group of Mr. Gerald D. Ojamar of Partido State University. So, ladies and gentlemen, this will be the order of presentation for this year's research presentation. I think without further ado, Ma'am Zell, I think we could already start the research presentation. Thank you very much.
Good day. I am John Patrick F. Pasibe, a fourth-year student of Bachelor of Science in Biology at the Partido State University. I will present to you a research study entitled Antimicrobial and Enzymatic Activity of Bacterial Endophytes Isolated from Malatungaw, Melastoma malabatrichum. According to Nair and Padmawati, 2014, endophytes are microorganisms that can be found within plant tissues without causing any harm to its host. Species composition of endophytes in one plant could also differ in its different parts, and these studies have shown that endophytes have greatly contributed to biotechnology in the production of biological active metabolites such as enzymes, phytohormones, nutrients, and minerals. These endophytes are considered beneficial in plant growth promotion, reduction of disease severity, and increasing plant mineral uptake, according to Melnick et al. 2000. These natural products could be harnessed for medicinal, agricultural, and industrial application. Stoma malabatricum L. Melastomata ca. is one of the 22 species found in the Southeast Asian region, including Malaysia. Considered as native to tropical and temperate Asia and the Pacific Island, this commonly found small shrub has gained herbal status in the Malay folklore belief as well as the Indian, Chinese, and Indonesian folk medicines. The objectives of this study aims to determine the antibacterial property of endophytic bacteria found in Malatungaw against pathogenic microorganisms. Specifically, it aims to isolate endophytic bacteria from Melastoma malabatricum growing in Barangay Maysalay, Goa, Camarines. To quantify antibacterial activity of endophytic isolates against selected pathogenic microorganisms, namely Staphylococcus aureus, Escherichia coli, and Bacillus subtilis. Qualitatively screen endophytes for their amylase and protease activities, and also to identify potential bacterial endophytes with pharmaceutical and industrial importance. The materials and methods that are used in this study are the following. Sample collection. 10 grams of healthy and mature stems and leaves of Melastoma malabatricum were taken in Barangay Maysalay, Goa, Camarines Sur. Samples were transported in a cooler and immediately processed in the laboratory. Isolation of endophytes. Surface sterilized leaves were cut into 5 mm square segments and were placed onto Petri plates containing nutrient agar, Na, for bacterial growth. Plates were incubated at 37 degrees Celsius for 48 hours. Following incubation, morphologically distinct bacterial colonies were isolated and purified on Na. Pure cultures were then qualitatively screened for enzyme activities. The water used in the final wash of the leaves was also plated in Na to test the effectiveness of leaf sterilization process. Antibacterial test of endophytic bacteria. Antibacterial activities against selected pathogenic microorganisms, namely Bacillus subtilis, Biotech 1679, Escherichia coli, Biotech 1634, and Staphylococcus aureus, Biotech 1582, were tested by agar well diffusion assay. Chlorampenicol served as the positive control, and sterile distilled water was used as the negative control. Antimicrobial activity was detected by measuring the zone of inhibition that appeared after the incubation period. Screening for amylase and protease activity. Qualitative amylase activity was based on King et al. 2014 with minor modifications for amylase activities. Skim milk agar was used in screening for protease activity. Pure isolates were point inoculated and incubated at 37 degrees Celsius for 24 hours. After incubation, zone clearing were observed. Identification of endophytes. The microcentrifuge tubes containing the cells of bacterial endophytes were sent in the macrogen for sequencing. Result in discussion. The sample collection and identification. The plant samples were taken in Barangay Maysalay, Dowa, Camarines. Samples were obtained near the river, rice field, and forested area. The plant samples were identified by the residences of the Barangay Maysalay and verified by the Institute of Biology, College of Science, University of the Philippines, Diliman. The figure shows the sampling site of Malatungaw plant in Barangay Maysalay, Dowa, Camarines. The figure shows the certificate of identification of the Malatungaw plant from the Institute of Biology, College of Science, University of the Philippines, Diliman. Endophyte isolation. Endophytic bacterial isolates were obtained from the leaves and stem samples. There were 5 samples of surface sterilized leaves and stem of Malatungaw plant, randomly cut and placed into the NA plates. After incubation, endophytic bacteria grew on the side of each strips and were subcultured to NA plates for purification. Selections of bacterial isolates were based on their morphological differences. Here shows the leaf and stem bits of Malatungaw after 48 hours of incubation at 28 degrees Celsius. Antagonistic activity. The test organisms used for the antagonistic assay were Bacillus subtilis, Biotech 1679, Escherichia coli, Biotech 1634, and Staphylococcus aureus, Biotech 1582. For the negative control, sterile distilled water was used and ciprofloxacin as the positive control. The figure shows the inhibition average of endophytes isolated from the leaves and test microorganisms. And it shows that the antagonistic activity of Delphia acidovorans, LA1, against E. coli and S. aureus as the highest in zone of inhibition. The figure shows the inhibition average of endophytes isolated from the stem test microorganisms. The result shows that Bromobacterium sp, SC1 has shown promising activity against Bacillus subtilis. Zymatic activity. Each isolate was screened for its ability to produce enzyme responsible for biomass degradation. Results show that 8 out of 12 isolates exhibited protease activity and 7 had amylase activity. This enzymatic activity were detected only for 2 strains from the genus Delphia and Cumamonas. The figure shows the qualitative plate assay for protease and amylase activity. Identification of bacterial endophyte. The cell sample of the bacterial endophytes found in Malatungaw was sent to macrogen in Korea for identification. The isolate LA1 were identified as Delphia acidovoran strain. The isolate SA2, SB1, and LC1 has the same microorganism strain, which is Delphia lacustris. The isolate SB2 were identified as Cumamonas testosterone. The isolates SC1 and LB3 were identified as Chromobacterium sp. Isolate SA1, LB1, SA3, LA2, and LB2 was classified as an uncultured organism, which may be due to the impurity of the isolated DNA. Conclusion. Endophytic bacteria were isolated from leaves and stems of Malatungaw, Melastoma malabatricum, found in Barangay Maysalay, Dowa, Camarines. Results of the antagonistic activity has shown that most of the bacterial endophytes isolated from the Malatungaw plant could partially inhibit test organisms. It has shown that isolate Delphia acidovoran strain LA1 had the highest zone of inhibition against E. coli and S. aureus, while isolate Chromobacterium sp LB3 shown promising activity against Bacillus subtilis. Results also showed that 8 out of 12 isolates exhibited protease activity and 7 had amylase activity and were identified under the genera of Delphia and Cumamonas. Recommendation. Based from the results obtained in this study, It is recommended to quantify the enzymes produced by these isolates and be characterized at the molecular level to further understand their mechanism of action. Some of the isolates were also not properly identified, which could be due to the impurity of extracted DNA. Thus, it is recommended to repeat the process DNA isolation and sequencing to identify these unknown isolates. It is also recommended to test endophytes against other pathogenic microorganisms, including fungi, to further know the scope of antimicrobial activity.
Okay, thank you for the first presenter. As stated in our guidelines, there will be a 5-minute time to answer questions from the panel of judges. So now we move on to the Q&A. Dear judges, you may now ask questions to our researchers, and to our researchers may we request if you can turn on your camera. Okay, so let's have first Ma'am Kathleen, or among the judges who wants to ask question. Okay, let's have Ma'am Marian.
Yes. Good morning. Can you hear me?
Yes po,
Yes,
ma'am.
ma'am.
Good
Loud.
morning po.
Okay. Good morning. Thank you very much for that very interesting presentation made by our young researcher. I have several questions, but in the interest of time, I will just choose the I think those that we need to address in the research that has been presented, research results. So more on the methodology, no? You mentioned that you have collected several samples or specimens of Malatungao in the different areas. Were there permits secured for the collection of these specimens?
Yes po, ma'am. May permit po galing sa barangay po ng pagkuha
Okay.
po namin.
Oo, because this is very important, no? In every bioprospecting research, it has to be mentioned not only in the paper but as well as during the presentation. So this will not be asked anymore. So second question is, um, did you perform the analysis of the sequences, or did you extract the DNA and then you submitted the DNA sample, or the pure culture? You submitted samples to Macrogen, am I right? Hello, can you hear me?
Yes po, ma'am.
Uh, uh,
Ang sinabmit po namin ay yung ano po, yung DNA sample po.
pure culture. So you were the ones who extracted the DNA. And then you submitted the DNA, and kayo din po yung nag-analyze ng sequences?
Hindi po, ma'am.
Okay. But I think it was not presented po, no, yung percent homology or yung match niya with those reported in the NCBI. Tama po ba?
Yes po, ma'am.
Oo, kasi you need to also present the results of the sequencing for us to really know ano yung percent homology niya with those reported, so we can verify the ID or the identification of your bacteria. And then, have you preserved and maintained these isolates or strains? Because sayang naman kung hindi nyo sila i-preserve. Are these properly preserved or maintained in your in the laboratory?
Yes po, ma'am.
Okay.
Naka-preserve po.
Okay. Ma'am, yun lang po muna. I'll pass I'll give the chance naman po for the other judges. Thank you.
Okay, let's have po Sir Edison.
Hi, thank you, John Patrick, for a very interesting research. However, yeah, to have a follow up on the matter on uh, the DNA extraction. Okay, so it was made clear a while ago that you yourself performed the DNA extraction, or what material did you send to Macrogen? Live uh, cells or just the genomic DNA, the total DNA of the sample?
The total DNA po, samples po, sir.
Okay. However, uh, it was not mentioned in the paper or in the presentation about the extraction procedure that you have performed. So as we know the methods. And aside from that, yeah, tama si Ma'am Marian. We need to check for the percent homology of the identity of your samples uh, to check the real identity na nakikita sa GenBank or sa NCBI. Kasi nung nabasa namin yung paper, you already claimed that these organisms belong to the particular species name, up to the species name pa nga yung sinabi. Minsan may 86% kaming nakita or even 70% or 76%. So we are uh, wondering if yun ba talaga yung identity niya. Perhaps a molecular or a phenotypic characterization like biochemical test or staining techniques could further support the claims. Okay, so we just do not rely on the sequencing, especially if these matches are very low, like for example 70 or 80. So that's really one of the help that we can get from um, phenotypic characterization. And last na, to give chance to Ma'am Kat, may I just ask what should be the purpose of testing uh, the potential of your isolates against Bacillus subtilis? So I understand that for E. coli is for gram negative, for example, or and for SRUs is a pathogenic organism that represents gram positive. What about Bacillus subtilis? As far as I know, and if my memory serves me right, I think there are no known pathogenic potential that can be uh, derived or can we know from the that species, or there are just one paper that say na pathogen siya, pero in general hindi talaga siya pathogen. So what should be the purpose? And yun lang. Thank you. Bakit sinama yung bacillus?
Sorry, sir. Yung ano po kasi nagpakita lang po siya ng promising activity niya po against dun sa test organism. Kaya yun po. Pero sorry po, wala pa po kami nababasa and will must get back to learn about it po, sir. Thank you po.
Okay, thank you, Ms. Kat.
Hello? Can you hear
Morning
me?
po, ma'am. Yes po.
Yes, good morning. Thank you so much for your very promising presentation. I really like it because it's very, sorry, andyan yung anak ko kasi. Wait lang. So, yun. I'm sorry. I have here my son with me. So anyway, yeah, your presentation is really, really nice. It's very promising. And in relation to what Ma'am Marian and Sir Edison suggested, regarding the preservation of your isolate, what kind of preservation are you using now? So because you said you have already preserved your isolate, what preservation technique did you use? Because it's really important to preserve it very well. Sayang nga, sabi nga ni Ma'am Maya, sayang pag hindi siya na-preserve. And yeah, just to answer the question, what preservation technique did you use? What method?
nakalagay po namin, ma'am, yung isolate sample namin sa ND ngayon at nakatago po sa ref ng lab namin po.
Oh, yeah. 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 That's but I would suggest that it's very risky, for since you are dealing with an identification study, and if you want to further your study, if you want additional studies to be done in your in related to that, it's very important to preserve your isolates very well. So I suggest that maybe you can tap someone from the biotech, I think, to preserve your isolates via freeze drying, because I think it's much better than just preserving your isolates in an NP. Also, we are uh, you may deal with problems regarding the passage of the uh, inoculating from the nutrient agar medium to another one. So it will affect the identity, because we know from the different studies that these microorganisms are very dynamic in terms of their microbial processes. So it would affect the identity. So another one would be more of the suggestion yung akin kasi. So another one would be as related to Ma'am Marian have mentioned, regarding the the molecular identification. So you didn't present the homologous, the phylogenetic tree that you maybe that could be another study to do.
Maybe, excuse me, ma'am. Maybe we will include the molecular identity in our final paper po. Hindi po kasi namin pala nalagay po. Thank you.
Yes, opo. Pwede po yun. So you can present that just for us to visualize kung ano yung relationship ng mga species, because on the species level yung na-present ninyo with the other microorganisms. So kaya yun yung maganda pag may phylogenetic tree. Because in the paper, I think you have you said that you set the samples to macrogen, right? So after that, the sequences you have to annotate it, you have to clean it, and someone has to do the blast, and you are doing that, or if you are going to use that. So another one would be for future directions. You can do since that's why it's really important to isolate. I mean to really preserve your organisms just for the future studies. So another one would be do a consensus identification of the isolate. What I mean by that is do just not just rely just what like Sir Edison said, not just rely on the molecular identification, just also do on other types. So we have other identification techniques, and it's really one promising. I mean, it's really what several studies now deal with consensus identification. So you can also present that as one of your recommendations. Okay. So thank you so much. Those are just actually suggestions. So thank you.
Thank you po, ma'am.
Okay, thank you to our first presenter. We now proceed to the